Good morning, everybody. Can you hear me? Okay. So I'll try to go slowly because they have to translate. So just be collegial with our colleagues in the back. It's a pleasure to be here. I was in France, beautiful country, a week or so ago in the city of Tours, and I had a wonderful time there. So I want to just recognize my colleagues here, former graduate students, and a former colleague, Dr. Charlie Staples, who have helped conduct a lot of the experiments that I'm going to share with you. So I'll make a disclaimer here first. I'm going to show you a lot of data from the University of Florida, but it doesn't mean that those are the only data on the subject that I'm going to talk to you. It's just because of the time available, I cannot go through the entire literature on choline in bovine, okay? So just be aware that there are other experiments done in other places, but just for the sake of time, I won't be able to talk about that. So I want to tell you a story. I'll give you a brief introduction to choline and how it came about to become an important nutrient. I will probably skip a lot of parts here because it's already been covered today relative to what happens to the cow during transition. I'll tell you a little bit of how we think choline affects the ability of the lipid to traffic fatty acids and export. And then I'll go on to the very applied aspect, which is what are the effects of supplementing rumen-protected choline on production and health of cows. And then I want to touch on some dogmas. There's a lot of dogmas in science, and this is probably one of those. The response to choline relative to body condition score of cows. And hopefully, by the end of my presentation, I hope to be able to support this statement that choline is not necessarily an essential nutrient from the biochemical point of view, but it's a required nutrient that should be part of the diet of adult ruminants. Okay, so this entire story started actually here in France and Germany. The initial discovery of this fatty type molecule, which was a nitrogenous compound associated with fatty acids, basically phospholipids, was actually took place in France initially by a pharmacist. If you read the first reports of phosphatidylcholine, it came from studies done in France. But it wasn't until the process of discovery of insulin by Sir Frederick Banting, a Canadian physician and scientist, with his student here, Charles Best, who did the groundbreaking work of discovery of insulin using a dog model, a depancreatized dog model, in which they surgically would remove the pancreas of the dogs during the process of discovering insulin. So Frederick Banting and a Scottish colleague eventually went on to win the Nobel Prize of Medicine and Physiology for his discoveries. So in this process, you know, a lot of science is done by observation. When you have students who are very keen at observing things, good things come out of that. So Charles Best noticed that dogs, when they were depancreatized, they develop hepatic steatosis, so accumulation of fat in the liver. Okay? So when he became a scientist, an independent scientist working in the US and Canada, he did the initial work showing that supplementing pancreatic tissue to dogs that were depancreatized and eventually supplementing phosphatidylcholine or choline chloride, he was able to reverse the degree of steatosis of the liver in those dogs. Okay? So that was the initial work showing that supplementing choline could reverse the degree of fat infiltration in the liver. It took about 65 years for the medical literature to accept choline as a required nutrient in human diets. So today we have guidelines. In the US, we have recommended amounts, what's called recommended daily allowance for humans, and I'll show you some. In the ruminant literature, nevertheless, it took some time to understand that this molecule, this trimethylamine, which is choline, it's unfortunately extensively degraded in the rumen, as many compounds are. So Derek Lindsay from England did the very early work showing that most phospholipids in sheep, presumably in bovine as well, were synthesized through a pathway that's called salvage pathway, through transmethylation reaction, not from incorporation of choline into phospholipids from dietary sources. And then Rich Erdman at the University of Maryland confirmed his findings, or their findings, with duodenally cannulated cows. And what Rich showed was that more than 90% of the choline in feeds never reached the small intestine. Okay? So choline, when it gets to the small intestine, is transported across the epithelium by specific choline transporters. Okay? But he actually got degraded before reaching the small intestine. In fact, choline is degraded by microbes, and it's reduced all the way to methane. So very little gets to the small intestine. So the ruminant animal is dependent on a particular pathway that I'll show you. So here's the classic major pathways for choline metabolism in tissues. So depending upon what tissue you talk about, so here is the molecule of choline. It has 3 methyl groups. It has a nitrogen. And if absorbed and direct to neurons, choline can be acetylated. It can receive an acetyl group, and it can become acetylcholine, the famous neurotransmitter that's important for nerve impulse and propagation of the nerve impulse across neurons or in the neuromuscular junction, like in this example here. Or choline can undergo phosphorylation in this pathway that's labeled here because of the American biochemist that described it. It's called the Kennedy pathway. And it becomes what's called phosphocholine. And phosphocholine can be processed in multiple tissues to synthesize numerous phosphatidylcholines. It all depends on the combination of fatty acids that incorporate into phosphatidylcholine. Okay? And we know that phosphatidylcholine is the main phospholipid in tissues. So choline is important for synthesis of neurotransmitters, important for synthesis of sphingolipids, phospholipids, for membrane fluidity, for transmembrane communication. Or it can be oxidized to this molecule here called betaine, which is an osmoregulator in the cell. But betaine has the ability to donate methyl groups. Okay, so it's what we call a methyl donor. Okay, so it's an important methyl donor. And it participates in this cycle of the folic acid, homocysteine, resynthesis of methionine in tissues. Like in the human literature, there's a lot of work on this because that's a marker for cardiovascular disease. So under the action of this enzyme here, betaine, homocysteine, methyltransferase, a methyl group can be transferred to homocysteine to resynthesize methionine in tissues. Okay? And this is enhanced by folic acid. So in the presence of folate, this process is facilitated. Like for instance, women who are pregnant, they all should get about 400 milligrams of folic acid because of neural tube defect in babies. Yeah? So there's malformation in babies if you have changes in the so-called one-carbon metabolism, which is the carbon that's being transferred as methyl groups. And then methionine can be condensed with another molecule called adenosine to form this other molecule here called S-adenosylmethionine, which is considered the major donor of methyl groups. Why am I telling you this? It's because methylation of regions of DNA in general suppress how genes are processed. They are transcribed. They actually silence genes. They can actually stimulate genes as well. Gene expression can be enhanced. But as a general rule, you see more silencing of genes, meaning that you don't produce the final protein of that particular portion of the genome. So acetylation, methylation of parts of the DNA or histones can alter how genes are expressed. Okay? So this affects the phenotype of the cell of the individual. But in our case here, the point is that S-adenosylmethionine can donate methyl groups to phosphatidylethanolamine that can result in the production of phosphatidylcholines in the hepatic tissue. So my point here is that in the ruminant, we are highly dependent on this pathway here for synthesis of phosphatidylcholine that's called the salvage pathway, because there's very little dietary choline to undergo the Kennedy pathway for synthesis of phosphatidylcholines. Okay? However, in the baby calf, when we feed milk or milk replacer is enriched with choline chloride, or if we are able to supply choline that escapes the rumen degradation, we can enrich this pathway here for synthesis of phosphatidylcholines. Okay, so how much choline do we need? Okay, Dr. Clay Zimmerman in the back there requires 0.5 gram a day. Okay, if you're a woman or a man, depending upon your age or physiological states, anywhere from 400 to 550 milligrams per day. Okay, poultry, depending upon the physiological state, require different amounts of choline. A lactating sow requires a large amount of choline. Okay, and because the quantities are large, we call the choline, it's kind of a wrong concept, a quasi-vitamin. You know, vitamin, if you read the literature, it's a vital nutrient requiring this very small quantity, micrograms, milligrams. Obviously, here we're talking about grams. But uncertainty is in the case of adult ruminants. We really don't know how much they need. And I'll tell you my interpretation of why we don't have softwares and models that tell you feed this many grams. I think there are two major reasons. We don't know the underlying reasons. Probably we don't know how much gets truly absorbed. It's not an easy task to do this. Choline is converted into multiple metabolites, and during absorption, the first pass through the splanchnic tissue gets altered, transforming beta-in and many other molecules. Okay, so we're depending on rumen-protected products that may have different bioavailabilities, and you will see it today later. And we don't have those response experiments that have titrated exactly the optimum. So empirically titrated the optimum amount of choline. I think that's why in the United States, the most recent guidelines called the NASCENT 2021, we don't have a particular amount of choline to be fed. And I think that's true for all methods in the world for guidelines of feeding dairy cows. So as many nutrients, the concentration of phosphatidylcholines in plasma, they go down around calving. Okay, so the amount of phospholipids in blood goes down around calving. My interpretation of this data is because both the pregnant uterus and the mammary gland are extracting those phospholipids for tissue of synthesis. In the case of the lactating mammary gland for synthesis of milk fat globule, for example, okay, associated with a reduction in supply of phospholipids because either there's less absorption of choline or there's less availability of methyl donors such as methionine, for example. So we are in the short supply here, relatively speaking, yeah? Now, this has been already covered, so I'll quickly go through this. I guess my picture of the cow didn't show up there, but there is a cow here. It's in there, okay? So I'll quickly go through this. So cows lose tissue at the onset of lactation. Mammals do that. It's perfectly normal. Probably the most extreme would be marine mammals, okay? So in this process of losing tissue, they mobilize fatty acids. They are used as energy source or as building blocks by the mammary gland to synthesize long-chain fatty acids. So for some of you who do farm work, you obviously know that your very first milk test has high fat content. And that is altered in terms of long, mixed, or short-chain fatty acids because there's a lot of C16, C18 fatty acids in milk at that stage just because a lot are preformed incorporated fatty acids from mobilized tissue, okay? Or they can be used by other peripheral tissues than the mammary gland, one being the liver. So this has been extensively covered. I'm going to just skip through this. They can be oxidized to ketones. This is actually a good thing, my opinion. I think in veterinary school we teach students wrong that ketosis is a disease. Ketosis is a mechanism of preserving nutrients biochemically. But obviously, if a cow is losing excessive amounts of weight, that's a bad sign. So ketosis is a marker for excessive fat mobilization. But ketones are water soluble. They do not require albumin to transport. So if this cow is not eating enough, her blood albumin is low, now you can transport carbon in a water soluble form. Ketones can be used to synthesize back acetyl-CoA, which can be used by other tissues such as the brain. Okay, so there's lots of good things about ketogenesis, although it is a marker for excessive catabolism. Okay, and this is a source of energy for synthesis of short-chain fatty acids in milk fat, or can be used by other tissues. Eventually, if you think about ruminants, they evolve consuming primarily carbohydrates and fermenting that into short-chain fatty acids. So the ruminant liver was never a lipogenic tissue. So never had to evolve exporting triacylglycerol like my liver, your liver, because we'll eat a meal, okay, we absorb glucose and other metabolites, and our hepatocytes will synthesize triacylglycerol and will export that as lipoproteins. But the ruminant liver didn't have to do that. So in terms of evolution, it's a somewhat different metabolism. Because of that, when you look at studies comparing secretion rates of VLDL, which is the lipoprotein that is exported by the liver to carry cholesterol esters, cholesterol, triacylglycerol, the secretion rates are much less in a ruminant liver compared to a monogastric liver. Okay, so the ruminant liver has, let's say, a harder time to export lipids, so relies primarily on oxidative pathways. So generate ATP for the cell, we'll use fatty acids or ketogenesis. Once the hepatocyte reaches a certain level of ATP, it doesn't need more oxidation, it doesn't need more energy, it diverts the pathway to ketogenesis. Okay, so it's a very orchestrated mechanism. Okay, so this whole concept of keep burning fatty acids to reduce fatty liver doesn't make biochemical sense. So there's no way to waste energy. But obviously this has consequences. It's already been shown here. It damages the tissue, affects the animal. The liver is a pretty important organ. Okay, so can we alter this? Can we alter this ability to secrete lipoproteins? So that's a question that's been out there for some time. Okay, at the same time, in early lactation, intake slow. Cows don't eat as much as we would like them to. That limits microbial protein synthesis. Okay? And microbial protein has about 2.4 to 2.6% of the protein as methionine. And methionine is this methyl donor that we just talked about. So we have low intake of escape amino acids. We have low synthesis of microbial protein. So all of that probably limits the supply of methionine to the cow. So some cows will develop fatty liver like this one here. We induce fatty liver in cows for experiments. This one here didn't make, as you can see. It's a very large liver. Liver of a cow typically weighs 9 to 11 kilograms. This was more like 18 kilos. So there's a lot of fat that accumulates. Yellow color. Can you see the yellow color? Yeah. So very strange. So how common is that? It's actually quite common in early lactation. So each dot here represents an average of multiple cows in the literature from the same shape and color represents the same study. They sample liver multiple times from cows in either on commercial farms or experiments. So yeah, you can see there is an accumulation of a triacylglycerol in the hepatic parenchyma in the first weeks of lactation. It takes some time until it comes down back to 2% or less. Okay? So we think when triacylglycerol is excessive, that alters the physiology of the hepatocyte. So how prevalent is hepatic steatosis? So here is data from England, data from other places in the world. Here is from the Netherlands. Here is from the US. And you can see that there's variability. But if you were to consider triacylglycerol of 5% or more of the tissue on a wet basis, that affects about 40 to 50% of the cows. So in this data set, 50% of the cows have some degree of steatosis in the early lactation. And that is associated with increased risk of other disease. So here is a paper that we try to understand, is there a particular cut point for hepatic triacylglycerol that predicts performance of cows? We were not able to find the exact value, okay? But I'll give you just a hint of the associations. This is not cause and effect, as it was already alluded. So it's an epidemiological study, a prospective cohort study. But it makes the point that cows that have excessive triacylglycerol, they have greater risk of disease, which is highlighted in red. They have increased morbidity. They have increased risk of culling from their herd. So yeah, there are consequences. Whether this is a causal relationship, we don't know very well. Okay? So in this particular paper, we use triacylglycerol as a continuous variable in the model. So we test linear associations, quadratic associations, and in general, the associations were linear. The more the cow has, the greater the risk of a negative consequence. Now, why am I telling you this? Because choline, through the synthesis of phospholipids, has this ability that we call lipotropic effects, which is the ability to facilitate the transport of lipids. Okay? So here is the first, probably the first study in literature that I'm aware of with dairy cows in which feeding rumen-protected choline reduced the degree of hepatic steatosis. So in this particular experiment, they collect liver tissue when cows were fed ad libitum. And you can see that the concentration of triacylglycerol was very typical of a well-fed cow. So low, about 2%. And these cows were randomized to either be fed a diet without rumen-protected choline or with rumen-protected choline. Okay? And then they underfed those cows. They limit nutrient intake to about 35% of the energy required for maintenance plus pregnancy. So they basically starved those cows for a little over a week. And then they collect liver tissue again. And what they showed was that the concentration of triacylglycerol was substantially reduced compared to the unsupplemented cows. Okay? So we repeated that experiment, but we titrate different doses of choline ion. And I know the story on choline sometimes is confused in literature. So choline is typically fed as a salt. In this case, is a chloride, choline chloride, and it's encapsulated. Choline chloride on a molecular weight has about 75% of its weight as choline ion, 25% is the chlorine. And then you have a polymer, a lipid coating material on top of that. So here I'm talking about the ion choline, not the final product, not the salt choline chloride. So you can see that as we increase the intake of choline ion from 0 to almost 26 grams per day, when we induce cows to develop fatty liver, which is the black bars, the concentration of triacylglycerol in the liver linearly decreased, corroborating the previous work from Rick Grammer's lab in Wisconsin that indeed supplementing Rumen Protect choline reduces the degree of hepatic steatosis in the dairy cow under this feed restriction model. So we recently replicated this experiment with a slight twist. So we assigned 110 cows that were going dry. They were 232 days of gestation, and they just completed at least one lactation. Okay, so we assigned them to 5 treatments, 22 cows per treatment. So the treatments were 0 grams of choline ion. And then we had 2 treatments with a particular product that had what we called a low concentration of choline chloride in the final product, okay, at 2 doses, 12.9 or 25.8, okay? And then we had 2 other treatments that I called a product with high concentration of choline chloride and also at 2 doses. I'm going to skip the product comparison here, and I'm going to focus on the effects of 0 versus 12.9 versus 25.8. That's what I'm interested here for this presentation. So the way we did this experiment, it was what we call a randomized block design, and cows were exposed to 5 days of ad libitum intake, followed by 9 days of feed restriction to about 42% of the net energy required for pregnancy and maintenance. During feed restriction, we supplemented those cows rumen-protected methionine to supply the same amount of methionine that was supplied when cows were fed ad libitum, based on the estimation of duodenal flow of metabolized methionine. So basically, these cows here, using the NASEN, the US model, so I'm not sure exactly in the INRA French model what the values would be, but I'm assuming not very different. These cows had a duodenal flow of metabolized or digestible methionine of about 20 grams per day. These were dry cows. So when we feed restricted them, we supplied Rumen Protect methionine to match the 20 grams of metabolized methionine. Okay? So this is what we found. So here's the liver triacylglycerol content. It's expressed on a wet basis or on a dry matter basis. So bovine liver is about 32 to 38% dry matter. So let's just look at the comparisons here. So here's the effect of choline, is the control versus the average of these 4 treatments here. You can easily see that supplementing Rumen Protect choline reduced triacylglycerol compared to the control. Okay? And now the second comparison that I want to make a point it is the dose of choline. It's the average of the 12.9 compared to the average of 25.8. You can see again that as I increase the amount fed, I further reduce triacylglycerol in the liver. So if you look at the average of these two, is about 6.6%. The average of these two here is about 5.5%. So the greater the dose that we fed, the smaller the triacylglycerol. So if you look at now at liver glycogen, which is the carbohydrate store of the hepatocyte, we see a completely different picture. The cows that we fed the control diet had the smallest amount of glycogen. As we fed choline, that concentration increased. And if I feed more, which is the average of these two versus the average of these two, you can see that the concentration further increased. So glycogen, we don't know exactly the underlying biology. Our hypothesis is that the choline that we're feeding gets oxidized to betaine. And betaine is an osmoregulator of cells, helps cells maintain water inside of the cytoplasma. And when cells increase in weight, kidney cells and liver cells, they tend to synthesize glycogen. So we think that, at least in the rat model, that's what it shows. So we think that it's an effect of this increase in weight of the cell that's stimulating glycogenesis. But it may be that we have less glycogenolysis as well in those cows. So when we look at the profile of the hepatic tissue, we have a more favorable profile relative to composition. If you look at the ratio of triacylglycerol to glycogen, that ratio is much smaller in the choline supplemented cows than in the control cows. Okay, so showed you 3 experiments that sort of replicates the findings in the dairy cow. So in this particular experiment, we looked at concentration of analytes in blood. So here is during the, before the experiment started, the concentration of fatty acids in blood, the concentration of ketones in blood. When we feed restricted cows, obviously they lose tissue. They mobilize triacylglycerol from body depots, and blood NIFA goes up, ketogenesis go up. We do not think that supplementing choline per se suppresses lipid mobilization. Okay, and I'm saying this because on farm, veterinarians will want to feed choline, and then they want to measure ketones in blood to see if choline worked or not. You may reduce ketones because supplementing choline may have beneficial effects to health of cows, and then they suffer fewer cases of disease, and then ketogenesis may go down. But I think it's fair to say don't feed choline and expect that you're going to have a reduction in ketogenesis because there is no direct mechanism there. Everything that we've studied in the liver, looking at transport of fatty acids, we see no changes with supplementing room protect choline. So I just leave that to your interpretation there. But one thing that we found that was quite interesting, these are all the treatments with choline. We induce feed restriction in these cows that alters fat accumulation in the liver. And we know that excessive triacylglycerol accumulation, as it was already alluded, induces inflammation. And one of the responses to inflammation is production of what we call this positive acute phase proteins, one of which is aptoglobin. So you can see there's a nice increase in aptoglobin in these cows, but not in these cows. So we actually repeat all of those assays because when the student came to me and we sat down to look at the results, I said, Usman, this has to be wrong. It doesn't make sense. So we reran all of the assays, and that's exactly what happened. So our interpretation of this data is 2. One is that either the choline is enhancing the phospholipid pool, phosphatidylcholines, and they have anti-inflammatory properties. They actually attenuate the inflammatory cascade. Activation of what's called toll-like receptor 4 and the downstream effects are attenuated by phosphatidylcholines. So that's one possibility. The other one is that we have less triacylglycerol infiltration in the liver, so maybe less pro-inflammatory stimulus from the lipids that accumulate there. So we can't really sort that out. But that was a nice finding there. Okay, so now if you go through the human literature, there's lots of mechanistic data on how the liver processes lipids. There's a lot less in the ruminant literature, and there's a lot of conflicting data. But we know that to synthesize this lipoproteins to export lipids from the liver, you need to traffic triacylglycerol and fatty acids across organelles in the liver. Okay? So one of the proteins that does that is this protein called MTP or MTTP. So that's called microsomal triglyceride transfer protein. It carries lipids across organelles. And then we have this apolipoprotein B100, which is the main protein that is part of VLDL. And then we have this other apolipoprotein. So in this particular paper here, what the investigators looked at was they compared the abundance of those proteins in the liver of cows that had low triacylglycerol content versus cows that had hepatic steatosis. It's an association study. And what they showed was that cows with fatty liver, all of them had, or all the proteins measured were in less abundance when those cows had hepatic steatosis. So here we can't really sort out whether the fatty liver is suppressing gene transcription and translation of the proteins, so you get less of them, or if it is the reduction in proteins is causing fatty liver. We can't really separate that from the study. But if we go back to other data, this is laboratory animal data. We know that when we feed choline-deficient diets, we limit the ability of tissues to secrete lipoproteins. Here is enterocyte. It's intestine now. Enterocytes synthesize a protein that is very similar to this. It's called apolipoprotein B48. The only difference is that the transcription of the gene stops at this particular codon here. There's a stop codon there that stops the transcription, so you don't have the same sequence of amino acids. And that's why it's called apolipoprotein B48. So in this case here, they measure secretion of ApoB48. And this is the main apolipoprotein present in a different type of apolipoprotein, lipoprotein, called chylomicron, which carries lipids from the intestine to the lymphatic system. So what they showed was that in a choline-deficient diet, this intestine of mice secreted less apolipoprotein B48 compared to choline supplement mice, showing that the ability to transport fatty acids in the gut is suppressed when we have a deficiency in choline in laboratory animals. So we complete 2 experiments to address those 2 areas, the liver and the intestine, indirectly. So I'll show you some of this data. So here is the first one in which we fat-restricted cows, okay? And then on day 8 of feed restriction, we stopped feeding those cows. We gave nothing to them to eat. There's 33 cows here. And then we intravenously infused this detergent called tyloxapol. Some of you who are in research is also known as Triton. And this is a non-ionic detergent that coats the lipoprotein particle and prevents lipases from hydrolyzing the lipoprotein. So prevents what's called lipoprotein lipase, which is present in the blood vessels, from hydrolyzing the triacylglycerol that's part of the VLDL, for example. Okay? And I'll tell you why we did this in a minute. So then we took blood samples sequentially from those cows up to 720 minutes. We had 3 treatments, a control, and then we had two types of two products, but I'm going to focus on the average of these two here, which is supplementing 25.8 grams of choline ion. And we collect supramammary lymph from the cow 6 hours after thiloxapol infusion. So what this thiloxapol does, it coats the lipoprotein particle, and it prevents lipases from hydrolyzing the triacylglycerol that's inside of the lipoprotein. So the cow develops hyperlipidemia. The blood lipids keep going up. Okay? And we measure lots of things in those cows. So in those blood samples. But I'm going to focus on the triacylglycerol because this triacylglycerol is as nascent, very low density lipoprotein. And the question that we ask is there a difference in the secretion rate of triacylglycerol from the liver if we feed or don't feed choline in those cows? So this is what we found. Here is the triacylglycerol from VLDL. And here is the control cows. Here are the cows supplemented with Room Protect choline. So you can clearly see that in these cows, the rate of secretion of triacylglycerol from the liver is greater than in the control cows. Maybe it's easier to visualize looking at what we call the area under this curve here. The area under the curve of the dashed lines is much larger than the continuous line. So you can see here there was a 40 to 50% increase in the rate of VLDL secretion by the liver when we fed Room Protect choline to those cows. So this corroborates data from monogastric. So the cow, this is nothing new. It's new in the cow, but it's nothing new in the literature. So the cow has a liver that's less capable of secreting VLDL, but choline stimulates VLDL secretion, even in the cow. Then we investigate genes involved in this process. Okay? So I'll show you associations first, because we don't have time to go out through the transcriptomic work. But so each dot here represents a cow. So you can see that for this gene, this is choline kinase. This is a gene that phosphorylates choline to make phosphocholine, that very initial pathway to synthesize phospholipids. Okay? So you can see as triacylglycerol increases, we have less expression of that gene. This is another gene in the synthesis of phosphatidylcholine. Same story. Okay? So if I show you two other genes, this is the ApoB100. Same story. So this is showing us that cows that have excessive triacylglycerol, somehow they have lesser ability to assemble lipoproteins based at least on the messenger RNA abundance. Okay, so then we ask the second question is, does it change intestinal absorption of lipids? So in this particular experiment, we assign 33 cows to the same experiment design. We had 9 days of feed restriction. In the last 36 hours, these cows were fed nothing. So they had completely empty gut. Okay? So they were fed only 4 kilos per day, and the last day and a half, 0. And then we basically dosed them with 450 grams of long-chain fatty acids. It was primarily palmitate, C16. Okay? And then we took blood samples, and we took sequentially fecal samples from those cows. And we measure an internal marker, it's called indigested NDF, to look at the fecal output and then measure digestibility, how much the cow ate, how much she excreted in feces. Okay? So here is the blood triacylglycerol. Cows fed room-protected choline at 25.8 grams had greater concentration of triacylglycerol in blood after we dose fat. But this is the most important thing. Cows that were fed room-protected choline had increased digestibility of fat. So about 6 percentage points. That's apparent digestibility. Okay? So we don't know the exact mechanism. Our hypotheses are that either the room-protected choline is helping the integrity of the intestinal lining or is enhancing the ability to traffic lipids across the enterocyte because of the role in the synthesis of phospholipids, which are important for chylomicron assembly and synthesis. So yes, when we feed choline, we get more choline in the blood, we get more choline in the lymphatic tissue or fluid. So that room-protected choline is providing choline for absorption in the small intestine. Okay, so let me quickly go now to the very applied aspect. It's taking me a little longer because I want to speak slowly. Is that okay? Okay. So this is a forest plot. And the way you read this plot is I have a response here. In this case, it's energy-corrected milk yield. Okay? So it's a milk that's standardized to 3.5% milk fat and 3.2% true protein in milk. Each name here, it's a comparison that refers to a particular publication in literature. These are all randomized controlled experiments in which every experiment had 0 grams of choline as a control and some amount of room-protected choline supplemented to the diet. So there are 21 publications here and multiple comparisons because of the different treatments in publications. The central line indicates no difference between the treated relative to the control. So exact same amount, value 0. If the point estimate falls to the left of the central line, it means that supplementing choline reduced the response. Okay? If the point estimate, what we call effect size, falls to the right of the central line, it means that choline increased the response. Okay? It's not showing here, but there is a gray square, which is the weight of that comparison in statistical analysis. So more precise experiments have more weight. If I have an experiment with 200 cows that has more weight than with 50 cows, that has more weight than with 10 cows. And the horizontal line is the confidence limit for that point estimate, meaning that this point here has a 95% probability to be somewhere here. It gives you some confidence of that point estimate. So here is the overall effect. Okay? What is this effect? It's about 0.38 standard deviations. How much is that in actual kilos? It's a overall response of 2.2 kilograms more energy-corrected milk. That's what the literature is showing to us. Now, there is an important point here. Is this what we call I squared. This is telling you that the response is heterogeneous. Okay? There's variability in the response. So we explore that. What explains the variability in response? That's one point. But before I go there, I want to make a very important point here. In science, when we run an experiment, we in academia think that that's the most important experiment in the world. We think it's the ultimate truth, and that is wrong. But when many people run similar experiments and you have somewhat consistency in response, that's what we call external validity of the data. It gives you confidence that under multiple farms, multiple managements, multiple diets, okay, et cetera, now I have a feel for how this technology will work in my own farm or in my clients' farms. Okay? So if you see a single experiment, that is just an initial step. But when you see 21 experiments, and in general, most of them show a positive effect, that gives you a lot of confidence to apply the technology. So we explore what was causing the heterogeneity in response. The first one is logical, was the dose of choline. So let's just look at energy-corrected milk. The circles that fall on the 0 here are the controls, 0 grams of choline ion. Okay, and then you have the intake of choline by the cows in the multiple experiments. The size of the circle represents the weight of that comparison, the statistical analysis. More precise experiments have greater weight than less precise. And the circles that are connected by the dashed line represent the same experiment. Okay? So we test what we call the linear and the quadratic response to choline, and we only picked up linear effects. What does it mean? In this particular case, the response to choline was linear. The greater the dose, the greater the energy-corrected milk. So that was one of the sources of variability in that previous plot that I showed you. So if I feed more, or if when people fed more in these experiments, the increase in energy-corrected milk was greater. So that's the first point. The second important point is the relationship between choline and methionine in the diet. So we collect the data from those experiments and enter all of those data into the 2001 NRC model that's used in the United States to predict metabolizable methionine supply. Okay, so we enter the ingredients of the diets and we adjust for the dry matter intake of the cows for the different treatments, and we calculate how much metabolized methionine each treatment consumed or absorbed, and what was the percentage of the metabolizable protein represented by metabolized methionine. And this was one of what we call covariates in the model, something that we add to the model to explain the milk production response. And just so you have a feel for where the data fall, most experiments, this is the postpartum data, the lactation data, the metabolized methionine represent between 1.7 to 2.4% of the total amino acids that were digested in the intestine. There were a couple experiments here in which there was very, very high because they fed a lot of room protect methionine. But you can see that most data fall in here. Okay? So what did we find? Let's look at the energy correct milk again. So this line here represents the milk yield, okay, for cows that were fed 0 grams of choline ion, according to the metabolized methionine as percentage of the metabolized protein. Okay. This here represents the energy-corrected milk yield of the treatments in which cows received 24 grams of choline ion. So how do we interpret this figure? So let's look at the bulk of my metabolized methionine data. It's between this range here. Let's avoid this very extreme and this very extreme. So you can see that when metabolized methionine is low, the response to choline is very large. When metabolized methionine is high, the response to choline is still there, but it's attenuated, okay? Which makes biological sense, okay? Which was very nice to find this from empirical data that matches some of the biochemistry. So the response is still there, but it's just reduced. So what happens to feed efficiency? You get more energy-corrected milk per kilogram of dry matter, about 60 grams more for every kilogram of dry matter consumed by the cow. Any benefits to health? So the data set on health is never as complete as the data set on production. So we had 21 papers. Here we only have 11 papers that reported health. So we have a less robust data set. But you can see at least for 2 diseases, retained placenta and mastitis, we were able to detect a statistical tendency for a reduction when cows were supplemented with room protect choline. So it's showing that there is some indication that health is improved with choline supplementation. Now, an important point, there are 4 experiments today that this data have been replicated. This was the first one that showed that. Here's the second, and then probably Clay will show you one from Michigan State University and one from University of Wisconsin. Supplementation takes place from about 255 days of gestation to 21 days postpartum. And you can see here that production continues to be greater for the treatment that receives room protect choline in early lactation. Okay? So it's a consistent carryover effect. At least 4 experiments show that. So what's the mechanism there? We don't really know the exact answer. It may be related to health of the cows. Healthier cows will have more production continuously. Or it may be related to our hypothesis is that choline kinase, this enzyme, is a marker for breast cancer in humans. And activation of choline kinase stimulates mitosis of memory cells. So we think that there's a possibility that supplementing choline enriches the choline pool of cells, okay, in this period here. And there's active cell proliferation in the mammary gland. And maybe that upregulates the activity of choline kinase, and that might stimulate more memory cell proliferation that results in this persistent increase, even when we stop supplementing room protect choline. Last point, what about body condition score of cows? Does it matter? Should I only feed to a particular group of cows? So we went back to our own data and asked that question. Is there a difference in response to choline, whether the cow is moderate body condition or she's over-conditioned? Okay. So here are the 2 experiments that we looked at that. This is the distribution of body condition score of cows when they enter the prepartum period. And the reason why I'm showing this is because in the US industry, there's been a revolution in this aspect. We have made major strides in reproduction that now we have to actually change the voluntary waiting period because our cows in the last few years have had superb fertility with a lot more milk. We go to farms producing 52, 55 kilos average, like 16,000 kilos. And they breed cows for the first time, and they get 55% of them pregnant the first AI with sex semen, something that we never thought would happen. So now we have lots and lots of farms that have shorter lactation and a lot of milk. So it's not uncommon for some of those farms to have cows with low body score when cows go dry. And if you think of choline, everybody associates choline with fat cows, fatty liver. So we ask, is there a difference in response if a cow has low body condition score? So the way we did this, we mathematically reanalyzed the data, and this is what we were looking for, so you understand. So in our mathematical models, we had the effect of treatment, which is room-protected choline, yes or no. And then we had the association with body condition score at the moment she enters the prepartum pen. And then we had the interaction between choline and body score. Okay? Each dot here represents a different cow. The blue represents the controls. The orange, the cows that receive room protect choline. There's about 190 cows there that were individually fed. So the question that we were interested in, is this interaction significant or not? And in science, we test hypotheses. Okay? And the way we test hypotheses, one of the things we look is this probability value that Sir Ronald Fisher in 1925 proposed. If this is less than 5%, you reject the null hypothesis and you say the means differ. Okay? If this is larger than 5%, you accept that there are no differences between treatments. That's somewhat subjective, yeah? But you can see here that these interactions have a large p-value, meaning that the response to choline, which is the difference between the blue and the orange line, is not affected by the degree of fatness of the cows when they enter the transition pen. Is that clear? So if you want to see, it doesn't matter if a cow was body score 3.0 in our scale of 1 to 5, or she was 4.0. In both cases here, she's producing another 2 kilograms of energy-correct milk.
So let me just summarize this whole story. Sorry that it took a little longer. This is a paper that just came in Journal of Dairy Science. You can probably find there very easily. If you don't, you can just send me an email or forward it to you. It's a review article on choline for cows. So what we think is happening, some of this is based on hard data, some of this is based on the best hypothesis based on laboratory work that we've done. Cows lose tissue to support the mammary gland in early lactation. A perfectly fine process. Nothing wrong with that. So some of those tissues in the form of lipids, fatty acids, are transported to inside of the hepatic cell as they are transported to many other cells. So there are transporters in cells here that facilitate the crossing across the hepatocyte membrane. If you have a very long chain fatty acid, 20 or more carbons, they usually are directed to this organelle here that's called a peroxisome. And peroxisome chops the fatty acid into shorter chains. And it does not produce energy, produce heat. And this generates the reactive oxygen species that were discussed before. But this long chain fatty acid can also be oxidized by the mitochondria. So depending upon how much ATP the cell needs, you oxidize more, you oxidize less. It's like, how much money do I need to pay my bills? I need to work harder or work less. I need more money or less money, okay? But if you get excess of fatty acids here, eventually they get packaged as what we call lipid droplets. And these lipid droplets are quite dynamic. They are not just storage sites. They secrete molecules. They change shapes. They are restructured, reorganized. And they can be degraded in this phagosomes or autophagosomes. And rumen-protect choline, at least at the level of gene expression, facilitates this process. We have 2 papers that have looked at that. So we think that part of the reduction in fatty liver is because of degradation of the lipid droplets in the phagosome of hepatocytes. But hopefully I showed you that supplementing rumen-protect choline facilitates the export of those triacylglycerols as very low density lipoprotein based on the rate of secretion in one of the experiments. I mentioned to you that phosphatidylcholines have anti-inflammatory properties, and I show you some data that it reduces this acute phase protein, haptoglobin. Rumen-protect choline enhances glycogen synthesis or reduces glycogen degradation in the liver. So the end result is that we have less lipids and more carbohydrate in the liver tissue, which is more compatible with a healthy hepatocyte. Okay? I showed you data that when we dose fat to cows, the digestibility of fatty acids is increased by rumen-protect choline. We don't know the exact mechanism, but I'll put forth 2 possibilities. Phospholipids are needed to maintain the architecture of the intestine. And when we underfeed cows, like we've done as our model, you know, that anxiety of the cow, she wants to eat, but I don't give her feed, that creates stress and that alters the tight junctions between the enterocytes, makes the tissue leak content, okay? And that affects digestion. It's possible that we enhance the architecture of the intestine to a more healthy type of enterocyte. The other possibility is that we provide phospholipids for synthesis of chylomicrons. And in mice, phospholipid deficiency makes chylomicrons to not be exported to the lacteals. Lastly, this is purely hypothetical now. We don't have data today to support this in cows. But I think this eventually is going to be shown. That's my take on this. Is that when we feed rumen-protect choline, we stimulate proliferation of mammary epithelial cells in that stage of gestation. So there's massive apoptosis of cells when the cow goes dry. And then about 2 weeks before calving, there's massive proliferation of cells to reestablish the mammary cell proliferation. So usually most of it happens in the last 2 weeks of gestation, and the first weeks of lactation. So we think there is a possibility for Rimprotect choline to stimulate mitotic activity, which would explain that increased persistence in production. Okay, so just to conclude, I hope that I made the case that choline fits this criteria of a required nutrient for cows. It improves health of the liver, reduces triacylglycerol, enhances energy-corrected milk yield, and has some indication to improve health of cows. At least we picked up 2 diseases that were reduced in incidence. The response to choline is dependent upon how much I feed and the metabolized methionine of the diet. The benefits are observed regardless of the degree of fatness. Skinny cows, fat cows respond to choline. And the response extends beyond the first 21 days of lactation. And not everything does that. And choline is one that does that. The mechanisms are probably multiple. We tend to focus on this fatty liver story because it's the most logical and the best known. But there's probably many other non-classical mechanisms. Okay? Reducing inflammatory response, memory cell proliferation, that might explain some of those effects. So thank you very much. Sorry that it took a little longer than I should. So thank you.